In this poster, we present a novel AI-based method to measure cerebellar volume change in multiple system atrophy, or MSA. The cerebellum atrophies significantly in MSA and can be measured sensitively using MRI. The work presented in this poster shows the validation of a new fully automated volume change workflow using the Jacobian determinant from nonlinear deformation fields estimated using a convolutional neural network, or CNN, from 3D-T1 weighted images. For the purpose of this work, we analyzed data from the MSTAR clinical trial in MSA, which consisted of individuals with both the Parkinsonian and the cerebellar subtype. In total, there were 191 participants, and 76 of those had the MSAP subtype, and 115 had the MSAC subtype. We did not differentiate between placebo and treatment groups because the labels weren't available. The dataset comprised of baseline and week 48 T1 weighted scans, which were pre-processed cross-sectionally and also longitudinally in a pairwise manner. The cerebellum was segmented at baseline using a multi-atlas parcellation method. In order for the longitudinal analysis, we trained the CNN to perform nonlinear registration of a finely registered baseline and follow-up image pairs. And this provided high-resolution deformation field maps in a fraction of the time usually required by other Jacobian determinant methods. Volume change measures were then obtained by integrating the log of the Jacobian determinant of the deformation fields within the baseline cerebellum segmentations. The three figures in the middle of the poster from left to right show the pre-processing workflow, the Jacobian CNN model workflow, and the Jacobian integration. Briefly, as part of the pre-processing, the baseline and the follow-up images are skull stripped, inhomogeneity corrected, and then aligned to the same MNI space. Then the follow-up image is registered to the baseline image, and both the intensity is normalized for both images. The resulting baseline and follow-up images are then used as inputs for the Jacobian CNN model. Once the CNN model performs both backward and forward registrations in order to ensure that the resulting transformations are symmetric and diffeomorphic, the output of the model is basically a warped follow-up image to the baseline and the deformation field that describes this transformation. In order to estimate volume change, the voxel-wise deformation field map is then summed within the cerebellar mask and then this produces cerebellar percent volume change measures. At the bottom, the last row of the poster shows the results of the validation, and the figure at the bottom left, we compared the CNN method against another alternative method for longitudinal volume change, which uses temporary coupled segmentation, and we refer to this as an atlas method. These both methods use the baseline the same baseline cerebellum mask. And as you can see, and we split it in red, it shows the MSAC subtype, in blue the MSAP. The CNN methods is able to measure sensitively the volume change in the cerebellum. For the MSAC group, it measures about 2.3% atrophy over 48 weeks. For the MSAP subtype, it measures 1.8% atrophy over 38 weeks. The atlas-based method is not able to capture any change in either of the groups, so both the estimated change is about 0. In the middle, the table in the middle shows across both the MSAC and the MSAP subtype the mean annualized change from baseline, which is about 2% for the Jacobian method, about 0 for the atlas-based method, and also compares it against the literature using the data reported for the PROMESA trial, the placebo group only. This kind of for this paper, they use the SPM12 approach, which is another Jacobian method. They report estimates of 3.4% with really high standard deviation, 3.7. The effect sizes for the Jacobian CNN method are really high, minus 1.6, whereas for the Promessa is 0.9. And using, assuming 8% power and a 25% treatment effect, I read a slowing of cerebellar atrophy by 25%. The required sample size per arm is 94 using the Jacobian CNN, or 300 using a method using the SPM12 method used for the Promessa. The atlas-based method reported change in the completely opposite direction to that expected, and pretty much almost zero change. So the sample size estimations are meaningless. So in summary, the developed framework provides a fully automated solution for estimating cerebellar volume change with increased sensitivity compared to other currently used methods. And in addition, this framework is really flexible and highly scalable and produces volume change estimates within a fraction of the time. It is therefore suitable for clinical trials, both large and small, in MSA and other neurodegenerative diseases where the cerebellum might be of interest. Thank you.